semen or saliva or blood from this possible perpetrator. And that's when we started with the touch DNA, because we know there was a violent crime, the victim was killed, and in Holland that often doesn't happen with knives or bats. We don't have so many guns. So in a lot of cases, there was contact between the victim and the suspect or the perpetrator, and we had to find this DNA. And then you come to the contact DNA.
Right. Now, you said that your methods were validated in 2002. Were these methods, or maybe I misunderstood you. I thought you said 2002.
I think that was in the national lab. So they did a validation study in 2000 already. And actually, when they were busy with the validation study, I used it in a high-profile case already. Oh. So from that time on, it was in the Netherlands. It was used in difficult cases, cold cases. I was project leader on cold cases. So yeah, I used it in a lot of my cases at that time.
My point, I guess the original question I asked was that the only additional step that you do is you just take additional runs of the PCR method to make additional copies. That's the, yeah. Is that correct?
No, I didn't answer your question correct, so I apologize about it. No, the problem, the point I wanted to make was that with the sensitivity of the techniques, it is possible to get DNA. It became more important to find the DNA. And till the day of today, that's still the problem. In a lot of cases, it's certain that the perpetrator had contact with the victim was dragged, but they don't find the location. So at that time, in 2000, it became clear that the technique was very sensitive, but you had to find it. And if you find something, or you had a location where you think, oh, this is a very likely location where the perpetrator touched it, you still, by a normal sampling method, you were not able to get a profile. So you had to play with the way in order to take a profile. For instance, over here I know the swabbing of clothing is a common sampling method. Well, in Europe this is hardly used because the results are not so good when you use swabbing. So we use a cutting method where you really pinpoint and you cut out a piece of sample and you dissolve the sample in the tube. And that's where we had a lot of success with cases, that we were able to find the right pinpoint location of DNA and then extract the DNA from that. But then, of course, the problem there is that if you have a piece of clothing, it could be that you need, if you don't know where to sample, you can take thousands of samples. And of course, as a state laboratory, you always are bound by certain amounts. You cannot do 1,000 samples on one item. So that was a big problem also in the national lab. You have to take the right position.
Well, my question really had more to do with the actual testing of the DNA, not the sampling. So I wasn't asking about the sampling, but the idea of running the test itself, it's simply a matter of running the PCR amplification for additional cycles.
For a low copy number, yes.
That's the only difference.
Sorry.
In that part. Okay. Now, let's talk specifically about a couple of the areas you've talked about. Counsel asked you about finding DNA in insects. Have you ever tested a particular insect called Megacelia scalaris, a fruit fly, a very tiny fly, mm-hmm. and successfully obtained DNA from that type of fly?
No.
You indicated in your testimony that those situations where getting DNA from an insect is successful are situations where the food, that is the blood or flesh, has been recently ingested. And that if it's recent, that sometimes you can still find some DNA that hasn't been digested yet by the insect, correct?
Yeah, well, I think I have to adjust that a little bit because the case we worked on and where we got the result was a 5-year-old case. So it's the remains where we got DNA profiles from. Whether or not at what time the insect died and or the pupae, the leftovers of the cocon, how you call it? Cocoon, where the insect is coming out. If they contain DNA, I don't know. But it could be in different stages. It could be that the insect or the larvae was dying at a moment, for instance by drying, and that the DNA stays in there then.
Okay. But you did say in your direct examination that it has to do with some recency between the consumption of the food and the test, because obviously, eventually, the DNA is digested by the fly and is no longer. Right.
If it's digested, of course, the digestion of DNA, the digestion of DNA would break down, and the further the digestion goes, the more the DNA breaks down. And we measure small pieces, but of course, also with UV light, for instance, if you break the chain, you cannot amplify it.
Yeah. So basically, even if you were able to obtain DNA from a fly as small as Megacilius scalaris, if it had been a month or more since it had consumed whatever human food it consumed, it would not most likely be available. It would have been digested.
If the fly consumed the blood or other cell material and then lived through and died at the end of the cycle, then the chance of DNA obtaining is of course small. If it died in between, before the consumption or the complete breakdown of DNA, then that's what I assume where we got the profiles from, is that, okay, then the digestion is halted, and then it's a way where the DNA could survive.
Have you ever done any published research studies on environmental effects on DNA?
I myself didn't.
You yourself?
No.
What is the most destructive environmental effect? What has the most destructive environmental effect on DNA? Number one enemy of DNA.
Well, it's a combination, I think, but moisture and temperature is a combination of moisture and temperature is a really detrimental effect.
So a hot, wet environment is the worst thing for DNA.
Yes.
And also the presence of bacteria is also extremely destructive to DNA, is it not?
Yes.
Would you agree, sir, that a piece of duct tape, well, let's talk about the duct tape for a minute. If you place a piece of duct tape on a person's skin and rip it off, it is likely to bring with it not only the dead surface cells, but the living sublevel cells, correct?
That's correct.
That's the point you were making on direct, correct? But when DNA is placed on a surface of the skin and not violently removed, then the cells that are in contact with the tape are, in fact, the dead cells, in direct contact with the tape.
Well, the dead cells, of course, the covering cells. Well, I think that's correct.
Yeah, the dead cells are the ones that are up against the tape.
Yeah, of course, depends how much force is applied.
Right.
And then but yes.
Okay. When it's torn off, it may pull with it some live cells, but what's in contact is dead, correct?
Well, in order to tear the other cells off, it should be also into the deeper layer, I think.
I mean, assuming it's not torn off.
Yeah.
That's my point. Okay. And you would agree with me, would you not, that duct tape placed upon the face of a human being who is then placed in a semi-swampy, occasionally underwater, hot environment in Florida, in an area that is an erstwhile trash dump, and sits there for 6 months as every bit of skin cell decomposes off the body, leaving it completely skeletonized. You would agree with me, would you not, that the likelihood of finding any intact DNA on that tape is extremely remote?
Well, of course, the conditions which you describe are very bad for DNA. This is not a condition where you would have your DNA in. But of course, depending on different circumstances, whether or not you lose all of it, as explained, we only need a smaller number of a small amount of cells in order to get a DNA profile. So the question then is, okay, would you lose all cell material? And that's possible, but it's also possible that still enough cells would be present to obtain a DNA profile, especially when using low copy number DNA.
But if there's no DNA on, when you test a bone for DNA, you don't swab the outside of the bone, do you?
No, if you want to identify the person of the bone, you cut out a piece of the bone or the skull. That's the two.
Scope. There was no bones brought up.
Because you don't expect to find any intact, recognizable DNA on the outside of the bone because it decomposes. Isn't that the reason?
The reason is that
Overruled.
Go ahead.
the body mass on the outside of the bones, of course, during decomposition has more, the DNA is degrading much more there. So why we use the bone and the skull is that because the chance of obtaining DNA profile from those locations is much bigger than from, and I'm talking about decompositional bodies which are in late stages, so there's no good blood anymore, and the skin and flesh are coming off the bones. That's the reason why we use it. Still, there is DNA in those locations, so it is possible, but it is way more difficult to get it from those locations, like for skin or the decomposing flesh or blood, than it is from the bones.
And while you have indicated that you believe it may be possible, you would agree it is extremely unlikely.
It will be very difficult to obtain a profile.
All right, let's talk about this masking issue that you discussed with Mr. Baez. You indicated that contamination in a DNA profile can mask existing DNA.
Yes.
But it would only mask existing DNA at exactly the same loci, correct? So going by your chart here that you use from your PowerPoint, the only thing that this 17 allele would mask would be a smaller 17 allele, correct?
This is not the profile that we were discussing.
It's an example,
Correct.
Judge.
Overruled.
Correct?
So, well, with the lower 17, it's like 55. No, that would not be very likely.
Right.
I think it was the other profile, the contamination, where.
Do you have a slide of that
Yeah, of course.
profile? Put that up, and we'll talk about that.
So
Let me clear this.
this is a profile where
This
there's
one.
more information visible.
Let me ask you the question then. Let's look at the various loci that we see here. Taking this 15 loci here, the only thing that would mask would be another 15 loci, correct?
That's correct.
It wouldn't mask a 14 loci.
No.
The same thing with the 16. It would only mask another 16. It wouldn't mask a 17.
No.
And the same could be said for each one of these loci. It would only mask its identical loci, correct?
Correct.
So in order for this profile to mask the profile of someone else, that person's profile would have to match at every single loci.
Yeah, it depends. What you see with the 17, for instance, what I already said, it would not be very likely that this profile is covering a full profile. So you will not lose a lot of information. What it would mask is, of course, the locations where you have the detection already, which you just circled.
Right.
And it's not so likely that a full profile would have been covered here. What could have happened is like a 17, well, in this case, there's no 17, but like a 16 or 15, if you have a very partial profile caused by a very small donation of a other party, that could have been covered. So then you would not expect that this covers a full profile.
Right. There's no way this covers a full profile.
No.
This masks a full profile.
No.
The only thing it would mask would be identical alleles.
Lower, of course, you see some alleles on this plate which are not mentioned, which are not detected by the threshold or by the analysis software. So Therefore, you don't know completely if there's all these alleles. And also, if you look at this, this would be a good profile to enhance with low copy number DNA. Then maybe there would be extra information. So yeah, we would use this as a start for the investigation. Say, okay, maybe we should do low copy number and see if we get more information there. Also, maybe with a third person. If you look behind the 15, there's a very small peak, but with low copy number, it's possible that you get a 16 there.
I'm sorry, you're talking about this?
The peak behind. No, that's in front.
I'm sorry.
That would be a stellar peak.
Hold on. Show me what you can illustrate with yours on the bigger screen here. If you use your finger, no, the one right in front of you. If you use your finger, you can circle or illustrate what you're referring to.
Oh, okay. Oh, sorry, I'm not so good at this. No, behind the 15, you see a small peak, and of course.
When you say behind, that's where you're losing me. I'm not sure what you're talking about behind.
After? Just if you have the 15. Where...
Talking about, that didn't work. Hold on. Let me get rid of this.
Sorry.
Okay, so just kind of put a little spot on what you're talking about.
Yeah, maybe I should just put that.
Is it where you have your, that little peak back there?
The second blue one, there's a small peak above that.
Well, there's a lot of small peaks in here, right?
Yes.
And you're saying that some of that could be noise.
Well, if you use the low copy number technique, that could be happening that you get information out of there. From this, it's correct, and the factory, I think, they use 50 as a cutoff for the analysis. Actually, the analysis is done by the computer mostly, and then later on, after the computer is finished analyzing, then a scientist comes and rechecks if all the data is correct or he agrees with that. So the data is correctly analyzed, but there's information there. If you look at these rounds, and these ones.
And what could those be?
So,
what could those be?
Those could be, I think if we use low copy number on these would go above the threshold and you would get information out of that.
What else could they be? If it's not DNA, what is it?
If it's not DNA. Well, these peaks can also be artifacts.
Okay. And again, your comments about what low copy number DNA could possibly do are completely speculative. You don't know whether it would come up with an additional profile or not.
Well,
Isn't that right?
pretty sure recognizing these locations. The peaks we see over here in the two circles are on locations where normally also markers are. So it's not pure speculation. That's that's the experience you need when looking over these profiles. Is that you recognize locations where you could get DNA extra information if you would use the low copy number method.
And every laboratory validates its equipment with certain limitations as to what will be called as an actual DNA peak versus what will not, correct?
Yeah, what I said here, it's the 50 RFU is a standard from the factory.
What is your RFU limits at your laboratory? Same?
We use the same.
So even in your laboratory, you would not call that as a DNA peak?
No, we would not mention this in the report, but we would enhance it.
You would potentially run a couple more runs of
Yes.
PCR?
Yes.
And then if it still didn't show anything, you would call it as nothing?
No, if the computer and the analysis software, which is highly sophisticated, don't mention it, no, we're not going to mention it.
Did you ask to retest this tape?
Yeah, we mentioned that we could investigate a lot of items.
But
Unregistered tape, yes.
Did you ask specifically to retest this tape?
Yes, we could retest the tape, yes.
It's not what I asked you. Listen carefully. Did you ask to retest this tape? Specifically this piece of tape?
To José Baez.
To anyone?
Anyone. Well, we mentioned that we could investigate this tape, yes. This piece of tape.
Are you aware if items were, at the defense's request, sent for additional DNA testing?
Object outside of the scope and outside of this witness's knowledge.
Overruled.
Do you know?
Well, yesterday during the deposition.
Well, other than what I told you,
Yeah.
because you can't really quote me.
Oh, okay.
Do you know?
Say move to strike
I just told him not to quote.
Objection overruled.
Okay. So aside from something I may have told you at deposition yesterday, as of yesterday, you have no idea if any item in this case was retested at the request of the defense.
No.
No further questions.
Redirect.
Mr. Eichenboom, you were asked about your laboratory being on a farm. Do you
Yes.
recall those questions? Your laboratory is actually known internationally as the crime farm, is it not?
Objection, Your Honor, as to what other people may call something.
Overruled.
Could I approach?
No.
Correct.
Okay. In fact, you've been sought out by other different countries to have DNA tested at your crime farm.
Objection, relevance.
Overruled.
That's correct.
And Mr. Ashton asked you about exposure. Your exposure, you've already had exposure internationally from your laboratory, have you not?
Objection, leading, and relevance.
As to leading, sustain.
Have you had any previous exposure just in the United States while you were in Holland prior to you being involved in this case?
Yes.
What kind of exposure have you received?
Objection, may we approach?
You may. Objection overruled.
Mr. Eichenboom, I want to talk about the recognition and exposure that you received prior to ever being involved in this case. When did you first start getting recognition from the United States?
Objection, it's not the question the court authorized him to ask.
Overruled. He can answer the question.
I can mention the name of the case. The first case was the Tim Masters case, which was an exoneration. We started with that case in 2006.
Okay, let me stop you there. What were you asked to do in that case?
We were asked to do contact DNA in that case.
And what do you mean by an exoneration case?
We were approached by a police officer who worked on a case, and she came to the Netherlands, or she was in the Netherlands on a visit, and asked, she visited us and talked about this Masters case, and she asked if we would be able and willing to do DNA in this case.
And was this to help free him from prison?
Sorry?
Was this to help free Mr. Masters from prison?
That's correct.
And what happened? What did you do?
We came over to the United States, talked with the defense team. Because there was no money at that time, we evaluated the case. I worked on quite some miscarriage of justice in the Netherlands, and thought, well, this could be a miscarriage of justice. So we decided to continue with the case. So, okay, we will, if the court allows us, do contact DNA in this case. And I testified about contact DNA in the case, and the judge allowed the evidence to go to the Netherlands.
And the evidence came to the Netherlands, and what did you do there?
We did do the DNA investigation, trace recovery, and DNA profiling. And then we were able to obtain a DNA profile, which was not matching the person in jail, Tim Masters. We obtained a lot of DNA profiles, all not matching Tim Masters.
Okay.
In 2006, there was not much interest, there was not a lot of media attention, but by the end of 2008, we were able to get a match with a suspect, or a former suspect in the case, more than 20 years ago. And that was the time that they released Tim Masters, and it was the first miscarriage of justice which was acknowledged in the state of Colorado. And by that time, there was a lot of media attention.
Okay. And you received international media attention as a result of that?
Yes.
And that was from being able to obtain a DNA profile from 20 years past on Touch DNA?
Correct.
Okay. And this person was found outdoors?
Yes.
And the clothing that you tested was subjected to numerous elements
Yes.
and held for 20 years.
Yes.
And then you finally were able to find something that wasn't found before.
Correct.
And subsequent to that, have you received other exposure from other cases?
Objection, leading.
Question is not completed yet. Overruled.
Have you received other exposure from other cases in which you have worked?
Yes. Because of this case, we got other cases from the United States, which also get some exposure in national media here.
Okay. And did law enforcement agencies, as a result of that, travel to the Netherlands to consult with you?
Correct.
Okay. And were you able to assist them in their cases?
Yeah, we were currently working on several cases.
Okay. So this case is not the first high-profile case you've ever worked?
No.
Okay. Now, when you talked about running extra cycles and that's the only thing you do, there are actually kits that are sensitive, that are more sensitive than your standard kits when running DNA, is there not?
That's correct. I had it in the presentation, but shortcut it.
Sorry, I didn't mean. I'm trying to keep it as simple as possible. Now, when we say kits, we're referring to the type of what is used to try and generate the profile, correct?
Yeah, you could say these are the chemicals used in order to, of course, it's a very complex procedure, but it's chemicals used to identify the different locations on the different chromosomes and make a profile.
Okay, and one example of this would be the mini filer
Yes.
kit.
The mini filer is specially made for broken down, deteriorated DNA, so that could have been helpful in this case.
And it's not just running extra cycles. There's also possibly extending the injection time.
Correct.
Okay, and that would, there are basically techniques that you use that might amplify the DNA or make it a little bit clearer so that the computer reads it. Is that correct? That's a poorly asked question, I realize, but I'm trying to.
Well, I think it comes back to what I already described. The trace recovery is very important. And that was underestimated in the 90s. Everybody was looking at DNA. But if I have a big stain and I just cut it out and there's so much oil or grease in it, or especially metals with the PCR, it could happen that there's a lot of DNA there, but you still don't get a profile. So you need to have a lot of knowledge about the amount from such a stain. You cannot just say, I cut out 10 by 10 piece of centimeters of stain. So cutting it at exact right location with the exact amounts of pollution, and also with DNA extraction, which I didn't talk about here as well, but DNA extraction, what kind of method do you use? The phenol chloroform will not be able to perform as good as a cleaning action as, for instance, the QIAGEN material. So certain filters, we use filters to clean the DNA, that would also make a difference in the chance of obtaining a profile. So it's the whole process from finding the stain till the end, or till analysis, in order to gain success with the DNA.
Now, and another reason that you are able to generate profiles where other labs do not. Also,
assumes a fact which is not in evidence.
Sustain.
Move to strike.
another thing that you
Just a second. Motion to strike granted.
Another thing that you do differently is, or another consideration is also the type of machine used to run a profile, correct?
That's correct.
Now, I know you have a slide for that. Can you bring that up and explain why that might help you generate profiles that other machines do not?
DNA is, can everybody see this?
Yes, yes, may we judge?
That's the same demonstrative exhibit number what, Madam Clerk? Okay.
So we see two machines over here. The one is slightly bigger than the other one.
They look the same size to me. Which one's bigger?
The right one is bigger.
Okay.
That will become visible if we look more closely. This is the 310, which we use. Is a smaller machine, and it's just one capillary.
And that's this line right here, correct?
That's correct. There's
Let
a
me just
line.
clear that out.
And actually, this is a very small tube in which the DNA is separated. And then a laser detects fluorescent labels, which we put on the small pieces of DNA, which we saw with the PCR amplification. And when the laser is hitting the label, it will fluoresce in different colors if it's a different marker or different location on the DNA. If you go to the bigger machines, they have 16 capillaries there. So now the laser has to go to 16 capillaries.
And that's these
The right
things
one
right
here.
here, right?
So the ABI 310, you can see it in the text, and the ABI 3000 are different in the way that the bigger machine has more capillaries, and that means that at the same time, where we do one sample, they can do 16. Well, one can imagine that for bigger laboratories, the state labs and the national labs, of course, they have lots of cases. And
Objection, Your Honor, this is not within the scope of cross.
It is actually, Judge. Mr. Ashton discussed that the only thing that he does different is run extra cycles. I'm pointing out that there are significant differences.
so the choosing of the machine is also of importance in order to get a profile. And we really tested this thoroughly because we do a lot of counter expertise in the Netherlands. So we test laboratory, or we do counter expertise on laboratory, which use the, all the bigger laboratories in Holland use the ABI 3100, so the one with 16 or even more now. And we found out that extracts where those laboratories actually didn't get any result, we were able sometimes to get full profiles, partial profiles, but we get a lot of results. And the only thing different, because we're using the same extract because they made the extract, so the only difference in the whole method was the machine. So that's why how we found out that the machine makes a difference as well. And of course, it does make sense if the laser has to go to one capillary. This is a glass capillary. If it has to go to one, of course, it's more sensitive than it has to go to 16, because you can lose some power of this laser light going through. So the machine using that it's slow, so that this takes time, and that's why I say we have a turnover time, which is longer, because we don't do 16 samples at the same time. We do one sample at a time.
Now, Mr. Ashton questioned you about degradation of DNA and how water and heat are bad for DNA. Do you recall that line of questioning?
Yes.
Have you found DNA profiles, and do you have any examples of what you can show the jury of profiles that items that you've been able to obtain profiles in that have been exposed to these types of elements?
Do you want to see the slide?
Sure.
Yeah, in this slide you see three different kinds of pieces of evidence. You see a panties, which is more than a panties, a pantyhose.
Yes.
On the left side, left upper side, and that's a piece of panty which is more than 20 years old. And after 20 years, we were still able to get contact DNA from these panties.
Now, I'm a little confused because this looks like there's a lot of blood here. How do you know? I mean, isn't that just the blood the DNA profile that you got from the blood, or is that?
The blood was from the victim, and of course, and it's more or less a given in the homicides we work on, is that you always have a lot of DNA of the victim, and of course, during our investigations, we're not looking for DNA of the victim, especially not on the clothing of the victim. So we're looking for DNA of possible perpetrators, and in this case. Of course, and that's why the sampling is so important. If you take a stain straight into the middle of the blood stain, you will have an enormous amount of DNA of the victim. But what you need is the DNA of a possible perpetrator. So taking a sample on a location, for instance, where there's less blood, where you're able to still enhance a profile of a second donor, that will help in order to get a profile. And that's what happened in this case.
Okay. And what else do we have here?
The second is the right hand of a victim which has been lying almost 3 weeks in the water. And also in this case, we were able not only to detect DNA from the victim, which if of course also 3 weeks in the water is not a very good condition, but also DNA from a suspect, which was later convicted, based also on the DNA.
Okay, and what about here, the jacket?
And the jacket is more or less the same. This jacket was not the body, but it was worn by a victim, which was lying in the water for around 3 or 4 days. And we were still able to obtain DNA from a suspect there.
So while, if I understand you correctly, while these conditions certainly don't help,
No.
they don't preclude you from being able to obtain a profile. Is that correct?
That's correct. Very often the bigger labs or the labs say, well, this has been in the water, we don't investigate it. And that's where we say, okay, well, give the evidence to us, we will try to investigate it. And then we were able to get DNA from that.
Okay. Now, you had requested to do DNA on items in this case, did you not? You were willing and able to do it?
Yes.
And you were willing and able to do it pro bono?
Relevance.
Approach.
Judge, just to move it along. Yeah. And the only reason you did not do the DNA testing in this case is because the prosecution objected to taking it.
Your
didn't you?
Honor, relevance. Yes, please.
Okay, we'll take our mid-afternoon recess to 20 minutes past the hour of 3. Please remember all of my previous admonitions. You may be seated. Okay, objection will be sustained in the sense that you can question him about the question that Mr. Ashton asked about, did he ask to examine that piece of duct tape? I guess that was on the mouth.
Allegedly, sir.
Well, wherever it was.
Yes, Q62, 64 group of items. Yes, sir.
And as to the other issue that you raised at the sidebar about asking the court to take judicial notice of different testing, give me exactly what you want, because I am really going to need to research that because of the issue of burden shifting. There is a very delicate and fine line with that. And that's something that I don't want to have to deal with at the last minute, because if I get it at the last minute, I'm going to err on the side of being safe and disallow it. But I need plenty of time, because those cases are all over the board, and it depends on which group you have, and the cases have shifted from time to time, just as the composition of the Florida Supreme Court, or if you look at the District Court of Appeals, whichever panel you happen to get. So that is not a very easy legal issue to decipher. Some things are best left unsaid. We'll be in recess.
Yes, sir.
Okay, state recognized presence of the jury and the defense.
Yes, sir, we do.
You may continue.
Mr. Eichenboom, the 17 allele that was found on the sticky side of the duct tape, it was found at one marker, correct? The D3 marker?
Yes, 17.
Okay. And in order to include someone as a match, you have to have all 13 markers identifying that person, correct? For inclusion, I guess, generally speaking now.
No, what you normally do is you have a reference profile, and that in the United States it uses 13 core loci, profiler, co-filer, and ideally you would have a match on all those loci. Okay. With a partial profile or a very partial profile, as in this case, you can only compare a reference profile, complete reference profile, with the 17 or the locus D3 here.
Now,
and the amelogenin.
Now, you testified earlier that you believe that this was human DNA, correct?
Yes.
17? Okay. Now, for exclusion, to exclude someone, how many markers do you need that is different from that person's profile? Did I ask the question?
As I understand correct, if somebody doesn't have the 17 in his profile, of course, if you don't have the 17 in the row of numbers of the 13 core loci, well, then you cannot have donated the 17. Is that what you mean?
Correct. So if I am a 14, 15
yes.
at this marker, which is the D3, and a 17 shows up, could you exclude this person?
Yeah, you would not be the person who could have donated the 17 then, because it's not in your profile.
Okay. I have no further questions.
Recross. Thank you, sir. Sir, one of the challenges with, let me restart. Original DNA testing was done with a system called restriction fragment length polymorphism, correct?
Correct.
And that system required a fairly significant amount of genetic material in order to get a profile.
Correct.
Sort of a quarter-size spot of blood or a bigger
yes.
or a substantial amount of semen.
Yes, correct. So blood and semen mainly.
With advances over through the early 90s, this PCR approach became the approach used by most laboratories, correct?
That's correct.
Because it replicates even a small amount of DNA to make it larger, to make it visible.
Correct.
As that has happened, the concern for contamination has become greater.
Correct.
In fact, the smaller amount of DNA you're looking at, the more difficult it is to relate it to the crime itself. Isn't that true?
That's correct.
So in other words, that you might find a speck of DNA through even just regular PCR or low copy number doesn't necessarily mean it relates to the crime.
No.
So the fear for contamination is much greater.
Correct.
Now, in this particular case, and I'm going to... I think Mike will be able to pick me up. If you had tested the tape in this case, there are 3 possible results that you might have achieved. The first is that you might have found nothing. Correct?
Yep.
And if you found nothing, that would not be inconsistent with the tape being placed on the child prior to decomposition, would it? Because as you said, the DNA could have simply degraded and decomposed, correct?
That is possible.
Yeah. The other possible result is that you might have found Caylee's DNA on the tape, correct?
Yep, correct.
And that obviously would be pretty strong evidence that the tape was placed over her mouth before she decomposed, correct?
Correct.
The third option is you could have found an unknown DNA profile on the duct tape. Isn't that correct?
Correct.
And there would be absolutely no way to ever know whether that unknown profile had anything to do with the crime. Isn't that correct?
Well, I think the district attorney was hypothesis, what we call it working hypothesis, was that the tape was crime related. So what you do with the DNA testing is test whether or not you get a result, and you can substantiate or refute, find support or no support for a certain hypothesis.
But my point is, if you got a result, an unknown result, a result that didn't match anyone that anyone knew anything about, you would never know whether that DNA came from something related to the crime or some contamination, which is very, very possible with low copy number DNA. Isn't that true?
Okay, low copy number DNA is more sensitive or prone to contaminations. That's true. We see that in the case we work in the Tim Masters case, for instance, we had a lot of contamination, which makes the whole investigation even more difficult. Having said that, what we do now is, and that's actually what the FBI also did, is testing that against all the persons in which you know who worked on the case, the crime scene investigators, lab technicians, everybody had contact with the evidence, you have a profile from. And I think in the United States also you have this database of profiles, not only from suspect, but also from lab co-workers. At least in Holland we have that. So everyone working on crime scenes or working in a laboratory has to give his profile and will be compared to obtained profiles. So if you can exclude or there's no support for hypothesis that any of these co-workers contaminated the crime scene or the evidence, then of course it becomes more likely that it is in some way crime related.
Well, but because this test is so sensitive, you can't ever know whether somebody had contact with their wife and then got transferred DNA onto an item because it's so hypersensitive. You just can't ever really know. Isn't that true?
Well,
you never really know.
No, it was low copy number. It's an issue which is more common. Of course, the type of transfer you're describing now is a lab technician having contact with his wife and contaminating the DNA with the DNA of his wife, but then still you would expect more DNA of this lab technician than from his wife. So this is called secondary transfer. Right. It's all possible. I won't deny that's possible. Secondary transfer is described, and there actually is literature about it. But it is less likely than direct contact, and so contaminations are easier found, and we, in the case we work on, we regularly find contamination by police officers or lab technicians. But then we recognize those.
Of course, the problem with low copy number is you may find unknown DNA, and then you'll never know whether it's contamination or an unknown suspect. That's the confusing part.
It's possible that it is, that you don't find out.
In fact, that happens fairly frequently where you get an unknown profile with low copy number DNA, and you really don't know where it came from.
That's possible.
Could you pull up that slide that you pulled up with Mr. Baez, the one with the jacket and all that in it?
Demonstrative exhibit by the defense, number what, Madam Clerk?
CI. Thank you, sir. The first object in the top left, was that associated with skeletonized remains?
No.
The object to the right, that obviously is a human hand that is not fully skeletonized. There's still tissue there. Is that correct?
Correct.
And the jacket that we see here, that was that associated with fully skeletonized remains?
No.
No further questions? Oh, I'm sorry. I don't think that, I've just told that may not have been published, and that's my fault. Let me go through that again. I apologize, just so the jury knows what we're talking about. May I publish it? Okay. None of these three, just to sum up,
Yep.
were associated with fully skeletonized remains, were they?
That's correct.
No further questions?
Redirect.
Based on what you do, these issues, the fact that they were, whether it's skeletonized remains or not, have no bearing on whether you would get DNA or not, correct? An item is an item.
Yeah, if there's DNA in the item.
Like, for example, these panties, you're not testing the skeletons, you're testing the item.
Correct.
Okay. Just like duct tape is an item.
Yes, correct.
The fact that the remains are skeletonized have no bearing on the item, do they?
Well, of course, it can give problems, but the item itself, it can be investigated. So, and then the condition of the item is more important than the skeletonization of the body. So the condition of the item, of course, is important.
And Mr. Ashton's 3 scenarios also leave out a 4th important scenario, do they not? Number 4 could also be that this duct tape was not wrapped around Caylee's mouth. Isn't that not true?
That's correct.
And in fact, based on what you've testified already, you would have likely seen some DNA based on the scenario that the state presents, does it not?
Well, if there was DNA, if the tape was placed over the nose and mouth, and especially the mouth and the mucosa from the nose, then there would be, at least at the beginning, there would be DNA.
And Mr. Ashton asked you about contamination and how sensitive DNA is now. When you find the perpetrator's DNA on an item, and you've testified in your experience that that DNA profile matches the perpetrator or the suspect.
The suspect and then
Yes.
later convicted persons.
You have never gone into court and said, you just can't ever know.
In what sense?
Well, Mr. Ashton was questioning you about, you just can't ever know if someone's DNA is on an item relates to the crime.
Well, the final decision whether it's crime related is in our system up to the judge and in this system to the jury, of course. So no, we would never go further than there's support for hypothesis this DNA came from this person, and on activity level, so it could be transferred. There's support for the hypothesis it is transferred during the crime or during pulling down the panties, for instance. So that's where we would go with this evidence.
Right, the evidence is either there or it's not. You don't give any interpretations and say things like, you just can't ever know.
No, it's on a crime level, yeah, it just match or doesn't match, or it would support a certain hypothesis or it wouldn't support a hypothesis.
There's either DNA there or it's not. Correct? I'm speaking generally, I'm not.
Yeah, well, if you have alleles, there's DNA, and if you don't, then there's no.
No further questions.
Just one. Are you telling this jury that the proximity of tape to rotting flesh does not increase the likelihood that the DNA on the tape would decompose as well?
It depends, of course. If the tape was on the body, which is decomposing, that would increase the chance that the DNA on the tape would also decompose.
No further questions. Thank you. Oh, I'm sorry. I thought you were done. Apologize. Go ahead.
Of course, it's depending if it's not on the body or it's a little bit away from that, then of course the chance of degradation of DNA is smaller. Of course, the outside conditions are still bad for DNA. On the other hand, of course, we have the PCR methods, which is so sensitive. We only need a couple of cells to obtain alleles or profiles.
So it does make a difference if the tape or item is in contact with decomposing flesh. It does make a difference, doesn't it?
Yeah, in my opinion, it does.
No further questions.
Yeah.